And then you can visualize. IGV is the tool that is the most used for visualizing next-gen sequencing data. So it's made by the Broad Institute. Download and install it on your own computer. It's a Java-based tool that works also on Windows. It works everywhere. So you open it, it will take some time. If it doesn't want to run, it means that there's something wrong with the Java on your computer. You have to install or update Java.
And IGV is what is called a genome browser. So it's a tool that allows you to visualize a genome.
It can also visualize the annotation in the form of a GTF or a GFF file, and it can visualize next-gen sequencing data. So we are using it to visualize BOM files, so the mapping results, but it will also visualize VCF files, so files with mutations, and it will also visualize peak files from ShipSeq, so it can visualize a lot of different things. For human and mouse, you even have extra information that you can visualize. You can visualize conserved parts in the genome, you can visualize repeat regions, and you can visualize the GC content.
That means that you have to give it a genome. So if you used your own genome for mapping, then you might also want to use the same genome here inside IGV. How can you specify your own genome sequence? Well, you go to Genomes and you select Load Genome from File. And it will open a window where you can browse. And then, so if you have unzipped the file, you can use here this sequence.fasta file. It's the same FASTA file, the E. coli genome, that was used to do the mapping on Galaxy. So you select the sequence.fasta file and you click Open. Then you need to give him the BOM files. So what you visualize is the sorted BOM files. How can you open the BOM files? You click the File button here, and then you select Load from File. You go to your unzipped folder that you downloaded, and you will see that there are two BOM files here, and each BOM file has a BY file with the same name. The BY file is the index. You don't open the index, you open the BOM file. So I do the 33 first, and you should see it popping up here in IGV. This will only work when the BY file is there. So this is why you need it. You don't open it, but it needs to be there. And then you do the same thing, so exactly the same, for the second BOM file. And they will open here as two different tracks. So the BOM files are open now, you don't see anything. Why don't we see anything? Well, because we need to zoom in first. How can we zoom in? Here, the plus button. So you click it, and you will see this getting smaller. So I click again, becomes smaller and smaller and smaller. And at some point, it becomes small enough for me to see, there we are, to see the reads. So all these gray things that you see here are the reads in my BOM file. So, and once you zoom in a bit more, you will see that the reads sometimes contain these colored lines. The colored lines are the mismatches. Mismatch between the read and the reference genome. I zoom in even more and more and more and more. And then you see not only the colored lines, but also the fact that the reads are small arrows. They have a direction. And the arrows represent whether it was mapped to the plus or to the minus strand in the genome.